Guillaume, who's the Development and Marketing Manager for EMD Millipore. He's going to be talking to us about implementing clone generation in fast-track manufacturing of proteins. Guillaume.
Thank you, and thank you for giving me the opportunity to present this new activity that we launched officially yesterday globally. To start, I would like to present EMD Millipore, which is the life sciences division of the Merck Group, the Merck KGaA Group, the German Merck. Millipore was acquired 5 years ago by the German Merck and became this life sciences division. And it today provides expandables, consumables, equipment and products in general for research labs, for quality control labs, and especially for manufacturing facilities. And that's the reason why we are here today, as this Process Solutions division provides filters, resins, equipment upstream, for downstream, in order to support our customers for the production of their drugs. And we not only provide products for the production, but we also provide services in order to leverage the potential of our products thanks to an added value. And this clone generation service is exactly the way we want to support our customers for manufacturing. So I think the first important point for clone generation and for manufacturing in general is that the decisions that are made at the very beginning of the process of developing a drug are really critical for the last stage, which is the market access of the drug. If you make a bad decision at the very beginning, if you choose a bad cell line, or if you don't choose the right cell culture media for the production, you may face big issues at the clinical stage, or even for market acceptation of your drug after clinical development. So it is really important to make the right decision at this stage. Do you want to produce a biosimilar drug? The biosimilarity is critical, and then the titer will be critical in order to control your cost of goods for the further production of your drug when you will be on the market. Do you want to produce a new biological entity? The key trends are different. You don't necessarily care about the titer, because the cost of goods is not the first thing that you have to care about. The price will be driven by the cost of goods at the end of the process. But you may definitely need to control the way you develop your drug and the IP that you need for the production of your drug. If you make a bad choice at the very beginning with a cell line which is under patent, you may have to pay a lot of milestones, fees, during clinical development and for market access of your drug, which most of our customers don't necessarily have in mind when they make the choice of choosing a clone generation provider. So making these decisions at the very beginning of the process development is really something critical. So, as I told you, we yesterday officially launched the clone generation service, which is a way to provide you with a cell line for the production of your drug, being a biosimilar or a new biological entity. It's not a revolution, but given that we do that in a GMP facility where we are able to provide process development support, GMP production support as well, we provide GMP batches to our customers. This is a way to ensure that your cell line will be produced or generated thanks to GMP requirements. And I will explain that afterwards, but there is one critical point, which is the clonality of your cell line that has to be taken into account if you want to be successful afterwards for your IND submission or for your clinical development. This is something that we are very careful with. as we are in a GMP facility close to Bordeaux in France. So the way we deliver these clones for the production of your drugs are based on three main points: reduce your cost of goods thanks to high protein expression, reduce your timeline thanks to a good template that we use and which make us having no surprises when we develop the cell line, and most of all comply with the GMP rules, which is again something really critical. So we do that in a very basic way, which is first of all the transfection of the DNA coding for your drug in order that the protein expression in the cell line allows the production of your drug. This is not something revolutionary. People do that for the last 40 years, but we have to do that. Then we perform a first and a second selection step, which allows us to ensure that the clones that we are going to work with, the pools of cells that we are going to work with, are made of cells which are transformed or transfected with your DNA of interest. And then we demonstrate the clonality. After the pool productivity assessment, we demonstrate the clonality of the cell line that we are going to deliver to you. And this is really the critical step. The FDA defined a way to ensure that the cell line is clonal a few years ago. And to do that, you have to follow the ICH Q5D rule, which is defined and which most of the clone generation services providers don't necessarily follow. But today, the FDA is really careful with this rule. And we use the only tool that allows the demonstration of the clonality of the cell lines, which is this equipment, the Clone Select Imager, which allows us to see one clone, one single cell, in order to ensure that the cell bank that we will that we will generate afterwards, and that all of the process which is going to be developed, and even the GMP production, even the commercial production afterwards, are based on one single clone that was isolated one time during clone generation. And this is really critical. Afterwards, we perform a gene amplification, and we provide our customers with a pre-MCB, a pre-master cell bank, which can be used in order to generate a master cell bank before process development. Master cell bank being the first step of the GMP process development of your drug. So to go further after clone generation, which is, as you understood and as you know definitely, the first step of drug development and manufacturing, we also provide process development support, which is made of a lot of different steps, like, of course, master cell bank establishment and USP and DSP process development. We can then provide you with a drug substance that can be used for preclinical studies, even if they are not produced thanks to GMP rules. But we can produce at this step, we can produce your drug in a reproducible, with a robust process in a reproducible way, which allows the delivery of something that you will be reproducible to what you will have for your clinical studies. Then we formulate, of course, the drug. We provide a working cell bank, viral clearance studies, all of the GMP requirements that allows you to submit an IND in order to go to clinic. And most of the time, we go from the cell line to the clinic within 12 months if it's, I would say, a basic protein that we have to produce, which is good, which is really good. But we have people in our facility who deliver this kind of services for the last 25 years. A lot of experience on 200 different proteins up to now, and this facility was acquired 4 years ago, but there is a lot of experience, of skills and knowledge in our facility, which allows us to deliver this kind of services in a right manner. And we can then use this process in order to produce a GMP material for your clinical studies. We have one full single-use GMP suite in our facility in Martillac, close to Bordeaux, which is made of state-of-the-art equipment for the production of proteins and monoclonal antibodies. So we have bioreactors from 50 to 2,000 liters through 200 liters that can be used for the production of phase 1 or 2 clinical batches, but also for phase 3 clinical batches, and if required for commercial production of batches of the drug. And then we have DSP suites, state-of-the-art DSP suites, which can be used for purification and formulation of the drug. And then formulation, and we deliver the drug substance, which we can transform into a drug product if required, thanks to partnership that we have with several providers in the world. After that, imagine we provide you with a cell line for the production of your drug. We develop a process for the production of your drug. We provide you with GMP material for your clinical studies, and you are successful with your clinical studies so that you can access the market, and you want to build up your own facility in order to control your costs. We can support you also for that, thanks to our facility design and implementation program that we call EndtoEnd Solutions. So we can copy and paste the facility that we have close to Bordeaux in Martillac wherever you want, in order to allow you to be able to produce by your own your drug wherever you want in the world. So some of our competitors claim that they can deliver the same service, the same solutions for end-to-end implementation of a new facility. But there is a big difference between them and us, is that we've done it for ourselves, close to Bordeaux, as I told you, and we know how long it takes. We know how hard it is to set up a GMP facility wherever in the world. So we have the experience of setting up such a facility, and we can support you in a very pragmatic way compared to what can be provided by others on the market. Just a remark. So after the setup of this facility, I would like to conclude on 2 ways. First of all, remember that the decisions that are made at the very beginning of the process development are really critical for the end of the drug development and for the access of the market. If you choose a cell line and if you make a mistake on this, it can be critical for the last stage. If you choose a bad cell culture media and if the yield of your cell line is not optimized with this cell culture media, then it can be critical for the last stage of your drug development. We have several services which allows you to make the good choice and which allows us to deliver you with the really state-of-the-art association of the cell line, the cell culture media, and the feed that can be used for upstream production of your cell line. And we, of course, have DSP equipment and products that we can deliver and use for this production. But again, whatever you need for your drug development, if you are at the first step, make no mistake and ask us if you want a cell line which is really GMP and which is really efficient for the production of your drug. To finish on that, if you've been successful with your drug development, and we even did set up a facility for you 10 years after we delivered a cell line for you, we can also support you in any way. We have hundreds engineers in the world who can be involved in the improvement of your process, or who can be involved in the debottlenecking of your process wherever your facility is in the world. We have half of these engineers who are in Europe and North America, and the other half everywhere in the world, in Asia, in South America, and in Australia. So we can support you in any way, and if you need anything else, let me know. I can answer your questions, or even take your URS if you have URS. Thank you very much.